Good afternoon, everyone. This is YY based on the GeneTex Taiwan office. And thank you for all of you interested in the GeneTex COVID-19 webinar. And thank you for being here today. Today's topic is an overview of the scientific and the clinical aspects of COVID-19, along with an introduction to GeneTex SARS-CoV-2 and coronavirus catalog of reagents. And our senior scientist, Mr. Alexander Bo, who is a doctor of medicine from the University of Southern California School of Medicine, will introduce this topic today. Before getting started, allow me to kindly remind that please help silence your microphone and turn off your webcam. And if you have any questions in the middle of the speech, just feel free to leave your message in the chat box, which is in the bottom right corner. And Alex will reply together at the end of the presentation. Thank you, and let's welcome today's speaker, Alex.
Ying, thank you very much for the introduction. And I'd like to thank everybody who's attending today for your time. I know it's getting to be later in the afternoon for you and near the end of your workday, and I appreciate your attendance. So as Ying said, the topic of the webinar today is SARS-CoV-2 and COVID-19, a world problem, need, and world response. As Ying indicated, we'll give sort of an overview of various topics relevant to SARS-CoV-2 and COVID-19, specifically the origin of SARS-CoV, very briefly, the present global situation, a little bit about the biology of SARS-CoV-2 in the context of other coronaviruses, in addition, clinical aspects of COVID-19 disease, and then again, as Ying indicated, an overview of GeneTex's SARS-CoV-2 and coronavirus product catalog. So just to start off with a little bit of levity here, a little bit of humor. So we can see here that somebody who was on a desert island wanted to be picked up in February, not so thrilled about being picked up in April, which reflects sort of the change that our world has gone through. And of course, something that we can certainly relate to on the coasts of the United States is as beaches reopen, it really keeps in perspective how things have been, how this disease and virus has affected everyday life. So most evidence indicates that in terms of the world impact, it began in Wuhan, the capital of Hubei province in China, in December of 2019, when patients began presenting to hospitals with severe pneumonia of unknown cause. Most of the patients had had some contact with the Huanan wholesale seafood market, which also sells live animals. Now, after SARS, the first SARS back in the early 2000s, a surveillance system was set in place by the Chinese government, and this was activated with these new cases. And respiratory samples were sent to reference labs for further analysis. By December 31st of last year, the World Health Organization had been notified about this. The next day, the Huanan market had been closed, and by January 7th, a coronavirus had actually been identified. And as we'll see, it had greater than 95 to 96% homology to a bat coronavirus and greater than 70% homology to SARS-CoV. Unfortunately, there was exponential case increase, which is driven by Chinese New Year travel and human-to-human transmission. By January 23rd, the entire city of Wuhan, with 11 million people, had been locked down. This then was expanded to the remainder of the province. The epidemic doubling time was initially somewhere in the realm of less than 2 days. And that once it had gotten out of China, people had seen exponential increases in Italy, South Korea, and Iran. And so by March 11th, the World Health Organization had declared SARS-CoV-2, COVID-19 a pandemic. An issue that we'll come back to a little bit later in the talk, but I want to introduce now is this whole notion of, in talking about origins of where this virus likely came from. And the greatest similarity, as shown here in this similarity plot, is to a bat coronavirus known as RATG13 from the horseshoe bat. And there's greater than 96% sequence identity between SARS-CoV-2 and this bat coronavirus. But what you'll notice is that overall, though the similarity is very, very high, a great deal of variability in the S1 region of spike protein. And this was, as we'll see, this lines up with the location of the receptor binding domain, and specifically the receptor binding motif. A recent study has actually looked at this in terms of trying to determine where the virus directly evolved from. And there is some evidence there that one of the intermediate host of the virus, so it goes from, as we'll see, from bats to some intermediate host and then jump to humans. Many people thought it was an animal called a pangolin. Now, pangolins are traded and sold in these markets. The meat is considered a delicacy. The scales are used in Chinese medicine. But what seems to be the case, if you look at, if you compare SARS-CoV-2 to a pangolin coronavirus sequence that was derived from 3 pangolins who were ill with a respiratory disease and ultimately died, they developed this sequence from that, or this pangolin, from these pangolins, and they compared that to the bat coronavirus that I just spoke about, TG13, as well as indicated here at the red arrow, SARS-CoV. And what you'll find if you compare the SARS-CoV to the bat, you see that overall the similarity is very high, even more overall than the pangolin. But what you'll notice is the extreme level, the increased sequence similarity in the receptor binding domain of the pangolin more than the bat, and we'll explore this a little bit more. If you now look at the spike protein, what you'll see again is the greatest similarity overall, pretty much, is with the blue, which is the bat sequence. But then if you look at this region, which corresponds to the receptor binding motif, which is the region that actually is making contact with the human cell receptor, you'll notice the greatest similarity here is with the pangolin sequence in orange. So that SARS-CoV-2 is genetically overall to the bat coronavirus, bat-CoV RATG13 than the pangolin CoV sequence. However, the pangolin coronavirus receptor binding motif shares 98.6% identity with the SARS-CoV. So what this indicates is that SARS-CoV likely derived from multiple recombination events between the coronaviruses of bats and other wildlife. And when you think about it, that's kind of chilling. So if you look at a phylogenetic analysis, to put this more in perspective, what you'll see is that the SARS-1, so to speak, or the SARS-CoV genomes are here. If you look at this area here, which is blown up, you'll see that the closest overall similarity is with the RATG13 bat coronavirus. But next to that is the pangolin coronavirus. So the thinking right now is all 3 viruses, SARS-CoV, MERS-CoV, and SARS-CoV-2, probably all originated in bats, or most related to bat coronaviruses. And then there were these intermediate hosts. For SARS-CoV, it was thought to be these civet cats. For MERS-CoV, it was thought to be camels. And then there's question, but it's suspected to be these pangolins, even though that nevertheless may have involved multiple recombination events. So just to quickly summarize this first part, most evidence suggests that SARS-CoV-2 originated in Wuhan, China, associated with the Huanan wholesale seafood market in December of 2019. There are some other theories that may have involved some other transmission between bats and humans. Bat guano, for example, is used as fertilizer in more rural parts of China, or perhaps between some other interaction between bat and man. In terms of when the virus most likely jumped from the animal host to humans, some analyses put it back as far back as mid-September, whereas others put it late November or December. The SARS-CoV-2 genome is, as we saw, most similar overall to the bat coronavirus RATG13, though there is nearly complete identity to the receptor binding motif found in the spike protein of pangolin coronavirus. This might indicate multiple recombination events between coronaviruses in bats and other wildlife prior to the jump to man. And as we saw, the SARS-CoV-2 was declared a pandemic by the World Health Organization on March 11th, so we're almost 2.5 months into this. If we look at the global situation, where we are as of yesterday in terms of, in this case, confirmed cases worldwide, what we see is the United States has the most cases. And this worldview seems to give an idea, it kind of gives an idea of sort of the flow of cases. So from China, went to Europe, to the United States. Now we're seeing more and more cases in South America, and as we see also in Africa. If we look at the confirmed cases per capita, we can see that the countries that have been, in terms of on a population basis, particularly hard hit. As we know that Spain, Italy, certainly United Kingdom, Sweden were particularly hit early. Mainland China has, as we'll see, the number of cases have slowed down. We're seeing cases increasing in Russia, of course in the United States, and as I mentioned in South America, we see it in Peru and Chile. If we look at hotspots, and this is areas where cases are doubling fairly quickly, what we see is, again, South America is starting to see more and more cases, certain countries, in addition in Africa. Whereas other places, as we'll see, countries that were affected earlier seem to be decreasing in terms of the number of new cases. And that's what's indicated here. If we look at case growth rate, and this is just the part of this list is quite extensive for all the countries that are affected. But what can show up just based on what I can fit on this slide were some of the countries that have been particularly hard hit and that were hit early. So as I said, the United States, close to 1.7 million cases, with almost, and at this point it's at 100,000 deaths. We see Brazil is getting hit more, and we see the case growth rate is indicated here. And you see, by and large, the cases seem to be slowing down. But nevertheless, we know that Spain, Italy, less so, also Turkey, less so particularly hard. Mainland China, as we see, the first hit, number of cases has started to decrease. If we look at the global situation in terms of new reported cases by day across the world, as well as new reported deaths by day across the world, there's both what looks like very good news in terms of the decreasing number of reported deaths, and also some mixed news, is because we can see that clearly a number of new cases on a world view has tended to plateau, but you are seeing a slight increase here. Now, whether that is because of increased testing or the fact that countries are trying to open up again and get their economies started, and so you were seeing increased cases, new additional cases pop up, is the question. So overall, if we want to look at the key points about the global situation, so as of yesterday, there were more than 5.5 million people sickened by SARS-CoV-2 on 6 continents and at least 177 countries. There have been almost 350,000 deaths. Worldwide, the number of daily new cases appears to be slowly increasing, or after plateauing, with an increasing number of cases in Brazil, India, Iran, Peru, Chile, Mexico, and among other countries. The number of reported deaths is decreasing, so that's the great news. The concern is, as countries begin to open up, the risk of case spikes remains an ever-present threat. Overall, on the world, the United States has the most cases. And if you look at a map edit, of a map of the United States in terms of where most of the cases are, you'll notice a large number of cases up in the Northeast, particularly New York, New Jersey. There were some in Pennsylvania, but also in the South, in Louisiana, Texas, Florida, in the Midwest, Illinois, and Michigan, and certainly on the West Coast. Washington was particularly hard hit early on, and certainly in California. Presently, the U.S. has almost 100,000 deaths, but although it's thought, new studies suggest that this actual number could be close to 130,000. So it might actually be 30,000 higher. The return to normalcy and the long-term effects on countries and the world economy remain to be seen, but appear to be quite serious. If we look at the biology of SARS-CoV-2, what we know is that it's a coronavirus, of course. Which are coronaviruses are envelope positive sense, single-stranded RNA viruses 60 to 140 nanometers in diameter. The family is Coronaviridae. There are 4 genera, alpha, beta, gamma, delta. The gamma and delta genera infect primarily birds and some mammals. There are 7 coronaviruses, some alpha and some beta, that infect humans. 7 total, as far as we know. These 4 are most often cause mild respiratory disease, though also some serious lower respiratory tract disease in children, elderly, something like croup. But by and large, primarily more moderate disease. There are 3 lethal coronaviruses that I think we all know. SARS-CoV-2, the present one, SARS-CoV. Both of these are beta coronaviruses, and they belong to the subgenus Sarbecovirus. And then, of course, the third one is MERS-CoV, also a beta coronavirus, but belongs to the subgenus Mirbecovirus. I think many of you are familiar with the general schematic of what a coronavirus looks like. So you have the single-stranded positive sense RNA genome embraced by nucleocapsid protein, and in addition to that, there are 3 other structural proteins. There's the membrane protein, an envelope protein, and I think we all are very familiar with the spike protein. If we look, the SARS-CoV genome is almost 30 kilobases in length. And if we look at sort of I'll show you a more detailed version of this, but of the actual genomic structure. But what we can see is comparing among the 3 genomes of these 3 lethal coronaviruses, we see there's 79% identity between SARS-CoV-2 and SARS-CoV, but only 50% between SARS-CoV and MERS-CoV. This is actually what I wanted to show you. This is probably one of the more detailed versions of, or views, or schematics, if you will, genome. Again, just about 29.9 kilobases in length. We see the ORF1A and ORF1B region that codes for the non-structural proteins, which are involved primarily in replication, but not, and then there's perhaps some immune modulatory proteins coded here also. And then, as I mentioned, we have the 4 structural proteins, and then probably as many as 9 accessory factors, which seem to be involved, have a number of functions which are still being determined. Some of them may be to counter the immune system. Others actually are involved in getting the virus released from an infected cell. As a matter of fact, there's a very recent paper about this protein 3B that seems to show that it's involved in suppressing the interferon response. So just in summary, the SARS-CoV-2 genome appears to code for perhaps 29 proteins or open reading frames. So as we saw, there's 16 non-structural proteins, 4 structural proteins, and at least 6, or perhaps as indicated here, 9 accessory proteins. Now, to get back to something I brought up, we talked about a little bit earlier, and that is what is specific about the SARS-CoV genome that contributes to its notoriety and its pathogenicity. And what you'll see is that, again, coming back to this sequence analysis in the receptor binding domain, when looking at SARS-CoV-2, that bat coronavirus, and that pangolin coronavirus, what we see is this high similarity, almost complete identity between the pangolin and the SARS-CoV-2 genome, including the complete conservation of these 6 amino acids indicated here that are key for ACE2 binding. Whereas only one of those is found in the rat coronavirus, and actually only one of them is found in SARS. So again, this receptor binding motif within the receptor binding domain is probably circulating in various pangolin coronaviruses also. So in other words, there's been an optimization that this is in the SARS-CoV-2 receptor binding motif for the receptor ACE2 binding. What also is suspected is that, or what's also clear, is that with SARS-CoV-2, there's a 12 nucleotide insertion, which is specific to SARS-CoV-2, that introduces a polybasic cleavage site, which leads to, which generates this polybasic cleavage site, which leads to more effective furin cleavage. So furin is a protease, and it definitely affects infectivity and host range. There's also some thought, and this still, I think, needs to be empirically proven for SARS-CoV-2, is that the placement of this polybasic site is just downstream of this proline. And there's some thought that that may lead to some O-linked glycan residue deposition on these two serines indicated in the box and this threonine, but this, which may be involved in immunoinvasion. But as we'll see, that really hasn't been empirically proven yet. The key, I think, is to really remember is that there's this polybasic cleavage site and this optimization of the receptors necessary for binding to ACE2. If you look at the SARS-CoV life cycle, just to give you an idea of how the virus infects a host cell, it comes in through the ACE2 receptor, and then as we see, one of the co-receptors or cofactors is a serine protease called TMPRSS2. The virus is taken in, it is encoded, it's an RNA, it's a positive sense genome, so it can immediately be translated to make its replication machinery, and then once that's in place, a negative sense strand is made for the generation of a series of nested subgenomic positive sense messenger RNAs that code for the structural proteins as well as the accessory proteins. In addition, of course, additional full-length positive sense genome is made for packaging. Translation occurs of all of the components. It's assembled in the rough ER and Golgi complex and new. As this audience is aware, the key protein that people have focused on is spike. And as we know, the SARS-CoV-2 binds to the ACE2 through its S protein. And the type 2 transmembrane serine protease TMPRSS2 binds and cleaves ACE2 and activates S, which leads to viral entry, as indicated here in the schematic. Both SARS-CoV-2 and SARS-CoV bind ACE2, which differs from MERS-CoV, which binds another peptidase called dipeptidyl peptidase 4, DPP4. And to sort of reiterate what I said earlier, SARS-CoV-2 spike S1, the S1 region is this domain right on top of the spike trimer that actually contains the receptor binding motif. And this spike S1 has 10 to 20-fold higher affinity for human ACE2 than SARS-CoV. And what we see in this model here is that the SARS-CoV spike receptor binding domain at the end of the SARS-CoV spike protein interacting with human ACE2, which is, of course, the receptor. To sort of revisit this issue of glycosylation, spike is heavily glycosylated. But what's interesting about it is that SARS-CoV-2 has 66 N-linked glycan sequons per trimer, so 22 per monomer. There's 23 per monomer on SARS-CoV, so there's 18 it shares with SARS-CoV-2. But what was interesting, and this is based on a couple of papers from the Crispin lab, is that this coronavirus, this spike, does not have a high glycan shield density. So this spike is actually more susceptible, as is the case with other coronaviruses indicated here, and to both to a humoral response, and that's what seems to go along with what we're seeing in the early vaccine trials. So the bottom line is, and the good news is that the spike protein is actually vulnerable to humoral attack is indicated, which is in line with other coronaviruses, in contrast to the more heavily to a higher glycan shield density seen with more immunoevasive or evasion-strong viruses, sort of including Lassa, some of the influenzas, HIV, and SIV. So, just to summarize this section, SARS-CoV-2 is one of 3 beta coronaviruses known to cause severe and potentially fatal disease in humans. The genome size is almost 30 kilobases. Genomically, it shares more sequence identity with SARS-CoV than with MERS-CoV. Key genomic features include point mutations in the receptor binding motif that optimize ACE2 binding, as well as the introduction of this polybasic site of 12 nucleotides that leads to effective furin cleavage between the S1 and S2 subunits of the spike protein. In addition, it's thought that there might be some additional O glycan addition, but as we saw, there wasn't, based on various studies, there's either very little or almost no O glycosylation detected on spike. So this remains to be more thoroughly evaluated. The SARS-CoV-2 genome codes for at least 26 proteins, though there does seem to be evidence for as many as 29 overall. The S1 protein receptor binding motif interacts with ACE2 like SARS-CoV, and entry is aided by the serine protease TMPRSS2. To move on to the clinical aspects of COVID-19 disease, I think we'll start with just sort of a brief summary to set the groundwork for the future discussion. So as we indicate, SARS-CoV-2 is more closely related to SARS-CoV than MERS-CoV. The two of these are in the similar clade or the same clade. MERS-CoV is in a different clade. The animal reservoir is bats for all 3 viruses. As we mentioned earlier, the intermediate host was thought to be these palm civets. For SARS-CoV, for MERS, it's thought to be with camels. And for SARS-CoV-2, as indicated, it's still a question, but it's thought to maybe involve pangolins at some point. Between SARS-CoV and SARS-CoV-2, the same receptor, ACE2. For MERS-CoV, a different receptor, a different peptidase known as DPP4. Now, these 2 columns are the ones we really need to focus on. If you look at the case fatality rate, MERS was particularly lethal, 34.4%. SARS-CoV less so, 9.5%, and SARS-CoV-2, the case fatality rate was the lowest of the 3, so 2.3%. The key here is this R0 number. R0 is a basic reproduction number. It's sort of a measure of transmissibility or contagiousness. So if an R0 is greater than 1, the outbreak is expected to continue, and the magnitude of the R0, the actual numerical size, can reflect the magnitude of the outbreak. Now, what you'll notice is that MERS was actually quite low, 0.7. SARS-CoV was less than 2, 1.7 and 1.9. SARS-CoV-2 initially was thought to be 2 to 2.5, so still the greatest of the 3, but recent evidence has put it as high as 5.7. And so by way of comparison, the seasonal flu, the R0 is about 1.3. The 1918 flu pandemic was about a 1.8. Measles, which is, I think we all know, is extremely contagious, has an R0 of 12 to 18. Nevertheless, the key thing, and one of the reasons this has been such a problem on a world level, is this high R0 for SARS-CoV-2. If we look at how patients present, and this is, again, is a very evolving field, but in sort of the classic presentation, fever and cough were extremely common, particularly fever. Shortness of breath in more than half of cases, muscle aches becoming more and more appreciated as a symptom, sputum production, headache, and then of course this, anosmia, the loss of sense of smell and the disrupted sense of taste, potentially indicating neural involvement. This is becoming more and more appreciated as an early sign of disease. So in a severe case, the median time from symptom onset to the shortness of breath to hospitalization and to, as we'll see, ARDS, could be as little as 8 days. The median hospital stay for patients, for recovered patients, is about 10 days. And sort of the operating thought is perhaps 4 out of 5 patients show no symptoms or very mild symptoms. 15% have more serious disease, but don't require invasive ventilatory support. And upwards of 4 to 5% of patients have very serious disease, leading to oftentimes to ventilation, to the intensive care unit, or death. So patient characteristics in one study, 14% of the people who presented to the hospital went to the intensive care unit, 12% needed ventilatory support, and of the patients admitted, almost 21% died. And if they were on the ventilator, 88% of them died. The laboratory imaging findings are particularly interesting and give an idea to the pathogenesis of the virus. What you see is in patients is leukopenia and lymphopenia in almost a quarter of the patients. So low lymphocyte count, low white cell count, increased AST in the patients with admitted to the intensive care unit, increased prothrombin time and D-dimers. There's increased troponin indicating potential myocardial injury, and all had CT chest abnormalities. 98% were bilateral, and as we'll see an example of this in a moment, the ground glass opacity with subsegmental consolidation. And really the key here for this, as I think becomes more and more the case, is the rise in inflammatory cytokines, including IL-2 and, as we'll see, particularly IL-6, but a series of pro-inflammatory cytokines that can lead to the very severe clinical course that happens in many of these patients. So what, again, related to that, what are some of the complications that happen to patients who have more severe disease? And that is ARDS, acute respiratory distress syndrome, secondary infection, acute lung injury, acute renal injury, cardiac injury, shock, and as we're hearing more and more about, thrombosis and other vascular manifestations of the disease, of the virus. Treatment has been largely supportive. Diagnosis through RT-PCR. People are scrambling to find drugs and agents that could be used to shorten the course. There's been some success with a viral polymerase inhibitor called remdesivir. And of course, at this point, no indication for steroids, primarily through infected respiratory secretions. But there's also what appears to be an oral fecal result, because virus is found in stool samples. There could be some actual fomite transmission, as was seen with SARS-CoV-2, but it's still to be evaluated. And the incubation time is most likely somewhere between 5 and 13 days. Diagnostic testing has largely relied on PCR-based methodologies. One of the more commonly used machines in clinical settings is this Abbott's ID Now COVID-19 test, which gives a positive result in 5 minutes and a negative in 13 minutes. So there have been some problems with this recently. What is being appreciated as we're now, 4 months into this pandemic, is that there appear to be additional manifestations of COVID-19 disease that seem to show how multi-systemic this virus can actually act. And children who are thought largely to be spared a lot of the more serious effects of SARS-CoV-2 disease or SARS-CoV-2 infection. Some children have been diagnosed with known as multi-system inflammatory syndrome in children, or MIS-C. Children present with fever, abdominal pain, vomiting, diarrhea, rash, conjunctivitis, and low blood pressure, and also peripheral edema. So their extremities fill with fluid. And so what is often seen is multi-organ development and increased inflammatory markers. So again, this consistent thing here, this consistent issue of increased inflammatory markers. They had similar symptoms as sort of an atypical Kawasaki disease or toxic shock syndrome, and it can develop 2 to 6 weeks after COVID-19 infection. The major concern here is that it's untreated, it can lead to cardiac and major vessel inflammation. What seems to also be unclear or observed, but the reason is unclear, it's often seen in ethnic minority children. Now, if the child is brought in and treated, the treatment is actually quite good, which is plasma transfusion, so intravenous immunoglobulin, and also aspirin. In addition, what you're seeing in adult patients are higher instances of strokes and blood clots. In one study, almost a third of COVID-19 patients in the ICU suffered thrombotic complications. In line with this is something called COVID toes, which is a red-purple rash in younger, generally healthy adult patients. And it's inflammation reminiscent of something called chilblains, which is a painful inflammation of small vessels in the feet due to exposure to cold weather. In addition, what we can see, what's also seen is myocarditis, kidney damage, gastrointestinal symptoms, and skin or dermatological manifestations. The other thing that is seen is with this notion of silent hypoxia. So somebody with extremely low pulse oxygen saturation on their pulse oximeter, when they should be in distress and extremely short of breath, they actually look quite calm and breathing normally, and no one understands why this is. And then, as we're seeing now, particularly in patients coming off the ventilator, the ones that get off or have been in the intensive care unit, is this notion of delirium and hallucinations, which gets back to the possibility of infection or somehow effect on the central nervous system. So we can see that COVID-19 was initially seen as a largely a lung disease, but now it can be appreciated as a multisystemic disease. And this is a lot of data from a study of almost 45,000 cases in China. The bottom line is, if you look at the case fatality rate, you don't want to be a patient over 60 to 70 years old, or certainly over 80 years old. You don't want to be male, and you don't want to have any pre-existing medical conditions, because all of these situations lead to higher mortality. Certainly, more severe case fatality rates certainly go up at 60, but really the case at 70 to 80 years old. Men seem to be more affected by severe disease than females. This seems to be a consistent finding. The reason for this is unclear, and certainly pre-existing medical conditions put you at higher risk for death and severe disease. Cardiovascular disease, diabetes, chronic respiratory disease, high blood pressure, cancer. All put patients at risk. This is a very complicated flow chart for the putative pathogenesis. I think what has to be kept in mind here is that ACE2, the receptor, is widely expressed. The nasal mucosa, throughout the respiratory tract, the heart, the gastrointestinal tract, the kidney, and also the brain. So this may be what's the reason for the effects throughout the body. Sort of the classic progression is direct infection through an aerosol or fecal-oral, gets access to the upper respiratory and lower respiratory tract. There seems to be associated immune dysfunction, which leads to this notion of cytokine storm. These high levels of pro-inflammatory cytokines can promote shock and direct tissue damage. There's complement activation. In terms of the respiratory presentation, the cytokine storm, you can develop acute respiratory distress syndrome, which oftentimes leads to death. If we look at this notion of cytokine storm more in depth, what we see is, as I mentioned earlier, that SARS-CoV-2 activates both the innate and adaptive immune responses. As we noted, lymphopenia is common in severe disease. So the numbers of CD4, CD8 T cells, B cells, natural killer cells, and the percentage of monocytes, eosinophils, and basophils are all decreased. What seems to be seen in severe cases, and actually a predictor of more severe disease, is increased neutrophils, or something called the neutrophil to lymphocyte ratio. The higher that is seen is a predictor of more severe disease and worse outcome. What's also seen in cytotoxic lymphocytes is the expression of exhaustion markers like NKG2A. So the immune system is just becoming exhausted. The good news is that for the patients that recover, these numbers all normalize, and as we'll see, antibodies are acquired. Feng et al., in conjunction with other studies, has shown that SARS-CoV-2 could infect seems to have the ability to directly infect some macrophages. Macrophages express IL-6 that can cause lymphocyte apoptosis. and actually lead to an upregulated fast-dependent activation-induced cell death. So what you see here in this schematic is in an uninfected individual, if we're looking at, say, the lung, the pulmonary epithelium, we see normal numbers of lymphocytes, CD4, CD8 T cells, no symptoms. In moderate COVID-19, what you see is slightly increased cytokine levels in macrophages, IL-6, and you see a slight lymphopenia, but nothing severe. And this is associated with fever, cough, fatigue, and myalgia in sort of moderate COVID-19. In the most severe cases, you get this notion of cytokine storm. So large amounts of IL-6 and other pro-inflammatory cytokines that lead to lymphopenia, low white count, fever, cough, fatigue, myalgia, shortness of breath, ARDS, and respiratory failure, and multi-organ involvement. Again, if we look at this sort of schematically to sort of illustrate it in a different way, if we follow this, we have SARS-CoV-2 binding to ACE2, the two types of alveolar cells in the lung epithelium, or even along the gut epithelium. You'll see a diverse multicellular immune response, macrophages, PMNs, and of course T effector cells and other lymphocytes. If you get viral clearance, what you've seen is maintenance of this sort of diverse immune response. However, as we mentioned earlier, in the more severe case where you get hyperinflammation, this pro-inflammatory response, lots of release of IL-6, what you're seeing is primarily macrophages and neutrophils, PMNs, in the response with the lymphopenia. And what's been, as numerous studies have been done to look at for active agents, what's had some success are agents that actually block this pro-inflammatory flow, this cytokine syndrome or the cytokine storm. And at least there's more than one, and so people are looking at a monoclonal antibody called tocilizumab, which actually has been validated for cytokine release syndrome and rheumatoid arthritis and other rheumatologic conditions. And what it actually does is an IL-6 receptor blocker. And so it actually blocks this action of IL-6 and blocks and sort of tamps down this cytokine storm. And there's been some positive results of this antibody. As I said before, I would show you a kind of a classic CT for a patient with severe and characteristic serious lung disease. So this is a CT scan. Here's the right lung, here's the left lung. And what you see is that if you look at the white arrows, that's this sort of bilateral ground glass opacities. And what you also see here, that in multiple lobes. And what you also see here is thickness of the pleura, where these black arrows are indicating here. And what's really interesting, and I think really sort of important in terms of the whole pathophysiology here, is indicated by this red arrow. What you see here is what this is, this clear area here, is actually a terminal bronchiole. And the reason you see it, because normally it's filled with air, but what you see around it, which should be just clear, sort of radiolucent area, is socked in alveoli. So these are alveoli that should be filled with air. And what's happening is they're filled with fluid, waste, damaged dead cells, that kind of thing. So it actually looks radio-opaque. And so it outlines the terminal bronchiole. And this right here, and so essentially what's happening is there's no air exchange going on here. And so this is actually what you see in the most severe of lung disease. What clinicians and researchers have tried to do to save as many lives and help as many people as possible is try to initiate many studies using drugs that are already have acceptable safety profiles and try to repurpose them for use against SARS-CoV-2. So, for example, lopinavir and ritonavir are protease inhibitors directly for HIV. As I mentioned earlier, remdesivir is a polymerase inhibitor. I think it was initially developed for Ebola. And this actually has been having some positive effect for severe cases. Lopinavir, ritonavir, and some of these other certainly chloroquine and hydroxychloroquine, which function to increase the pH of the endosome and potentially block viral uncoding and processing so that it can express, so it can replicate, which has not, these have not been shown not to be effective and have actually shown to be potentially quite dangerous. What seemed to have been, what's the medications that at the present that seem to have been most useful are certain monoclonal antibodies and also convalescent plasma from people who have recovered from disease. There's also use of this, or actually people are looking at camostat mesylate, which is a serine protease inhibitor. As I mentioned, the TMPRSS2, that cofactor, is a serine protease. Camostat mesylate is actually in use in Japan for treatment of chronic pancreatitis pain, so it has an acceptable safety profile. So people are seeing if they can block infection or inhibit infection by using this, and so there are ongoing studies with that. And of course, new antibodies and neutralizing antibodies are of course being developed. As countries around the world see their COVID-19 case number plateau and then hopefully wane, what is next? And the thing that's being developed now, of course, or trying to be developed, are reliable antibody testing. And why is this important? To determine who is immune, and this is particularly important as countries try to remobilize their economies and their workforce. And of course, very important, most important for healthcare personnel. It's also crucial for being able to assess vaccine efficacy and also contact tracing, and also to inform public policymakers about the true number of asymptomatic cases. We still don't know across the world how many people have been truly infected by this virus, and it won't be known until you can come up with reliable antibody testing to detect SARS-CoV-2 specific antibodies. Of course, the primary target for these neutralizing or these antibodies is most definitely spike protein, but also potentially nucleocapsid and perhaps some of the other couple of the other structural proteins. One key issue, of course, is to detect antibodies that are specific to SARS-CoV-2. Many of us have probably been exposed to the other coronaviruses, which cause just cold symptoms in most of us. So that remains to be seen also. And the key question, of course, is immunity lifelong? Now, based on some of the initial vaccine results and some of the results with SARS, who are now 17 years out, there's some optimism that there could be some long, certainly lifelong or perhaps extended immunity from patients who have been exposed to SARS-CoV-2. As there does seem, based on evidence, there does seem to be both a humoral and T-cell based response generated. So that's encouraging. If we look at the vaccine development that's underway now, this number is probably low, but at one time there were at least 40 pharmaceutical companies and academic institutions working on vaccine candidates. And as we know, there are different approaches. There's at least 5 different approaches for developing vaccines. A live attenuated virus vaccine using viral protein injected, so a protein-based vaccine, combined with an adjuvant. A popular approach is an mRNA vaccine and a DNA vaccine, and then also a viral vector vaccine. How to assess the efficacy? Some are using mice that express the human ACE2, virus-like particles, and a good model that is being used to assess some of the vaccine trials are rhesus macaques, or macaques. And just so you know, the Moderna vaccine that's had some very positive early results is mRNA-based, while the Oxford vaccine is an adenoviral vector expressing spike protein. So clearly there's ongoing, there's intense work being going on in vaccines. Some people say a vaccine will be ready by the end of the year. Others might think not until next year, with 12 to 18 months, the usual target for vaccine development. So the key points for the clinical aspects of COVID-19: SARS-CoV-2 has a lower case fatality rate than SARS-CoV or MERS-CoV, but has a much higher transmissibility. Remember that R0 of 5.7. Clinical progression in serious cases from onset of symptoms to hospitalization and ventilatory support can occur in a span of 8 days. Male Men seem to be more affected with severe disease than females. Age is certainly a risk. Over 60 years, but certainly over 70 to 80 years, and with pre-existing condition puts people most at risk for severe or lethal disease. Pathogenesis involves cytokine storm, multiple organ development, ARDS development, though as we mentioned, most of the cases, perhaps as many as 80%, are mild or asymptomatic. And what's becoming more and more the case, it's SARS-CoV-2 disrupts the entire immune system and also targets vasculature. There's intense research in the identification of existing agents with acceptable safety profiles for use against SARS-CoV-2. At this point, certain monoclonal antibodies and perhaps remdesivir are leading candidates. As the pandemic eases in various countries, there'll be, of course, an emphasis on virus-specific antibody detection. So at that point would be IgG. And vaccine development is ongoing, although there's some really optimistic early results. I just want to quickly, just to wrap up, go over some of the products that highlights what GeneTex is doing in terms of its SARS-CoV-2 catalog to try to assist with the research effort against the SARS-CoV-2 and COVID-19. What you'll see is that at this point, and we even have more coming down the line, we offer multiple antibodies. I think there's at least 9 here against spike protein of SARS against SARS-CoV and SARS-CoV-2. In addition, 3 nucleocapsid antibodies, an antibody against ORF7A and NSP8, multiple antibodies against ACE2 and TMPRSS2. We also offer camostat mesylate for in vitro using, again, that serine protease inhibitor. You'll notice, just to sort of give an overview of some of where we target various antibodies against spike and nucleocapsid, that's indicated here. And so many of these products are already available or will soon be released for both targets, both spike and nucleocapsid. What you'll see on several of the slides is this red lightning bolt, and what that means is that these antibodies were actually tested on virus-infected cell lysates. So you see here some of the antibodies that I indicated earlier for Western blot. Some were actually raised against SARS-CoV, but actually have been shown to cross-react with SARS-CoV-2. And we see here them available and active in multiple applications. What we're excited about is GeneTex has just opened a new state-of-the-art recombinant antibody facility that generates rabbit monoclonal antibodies. And the advantages of these are high sensitivity, low background, batch-to-batch consistency, routine resupply, no animal usage. And we have 4 now available, and there are more coming down the line against spike S1. 3 at this point released for nucleocapsid, and there's 4 new ones, recombinants against nucleocapsid that are to be released very shortly. So that'll be a total of 7 against nucleocapsid. And then in addition to that, we offer recombinant proteins for SARS-CoV research, including nucleocapsid protein, different variants of the spike. We also have against envelope, against the NSP5, which is 3C-like protease, and then human ACE2. And then, so a total of 4 spike, 2 nucleocapsid, an envelope, 3C-like protease, ACE2, recombinant proteins. And then we also, as I'll show you, we also have recombinant proteins for comparative comparative reasons for MERS-CoV and SARS-CoV-2, SARS-CoV spike proteins. We also have cell lysates that overexpress spike, or it's the full-length spike, the S1, S2, the receptor binding domain, the nucleocapsid, and as I mentioned, that lysates that overexpress either SARS-CoV spike or MERS-CoV spike. We also are selling FFPE overexpressed cell block pellets against, obviously the structural proteins. These can be used as sort of an adjunct positive control for IHC, as more and more of our antibodies are tested for IHC. In addition to SARS-CoV, we also carry products directly for SARS-CoV. MERS-CoV. And what we're very excited about, and we're in the process of developing sandwich ELISA pairs. We've already identified at least a couple of pairs for spike, and some additional pairs are being tested right now. So that could be particularly used for the development of testing kits for in vitro use, of course. And then also nucleocapsid sandwich ELISA pairs also. So we're very excited about these sandwich ELISA pairs for both nucleocapsid and spike. And more of these pairs are being presently identified. To examine cytokine storm, what we have is an extensive line of reagents to study for immunology, and this includes key targets involved with components of cytokine storm. So IL-1, as we mentioned earlier, on IL-6, IL-10, CXCL8, GCSF, and this is just a few of the catalog. And then soon to be released SARS-CoV-2 products are a slew of new antibodies against envelope and membrane, so the remaining 2 structural proteins, several of the key non-structural proteins, NSP1, the 2 proteases, NSP3, NSP5, NSP12 is the actual polymerase, NSP4 is a methyltransferase and exoribonuclease, which is a key part of the replication machinery, and ORF8 and ORF10, which are 2 of the accessory proteins. We have new recombinant proteins that will soon be released, including membrane, the spike with the receptor binding domain with a flag tag, and NSP5. Overexpressed 293T slides lysates for envelope, nucleocapsid, various forms of spike, and as I said, new ELISA kits coming down the road. And I'd like to, acknowledge and thank all the local distributors throughout Europe and Turkey and Russia for the pleasure of working with you. Thank you very much.
Thank you very much, Alex, for this very informative presentation. So I just received that some feedback that it seems some customers' webinar platform is in Taiwanese. So we are really sorry for this inconvenience caused. So if you have any questions, the chat box is on the bottom right corner. But if it's still very hard for you to find, you just can email the questions to GeneTex or your local distributors. So our email address is info@genetex.com. Okay, so I have one question, Alex.
Yes.
So as I know, GeneTex conducts a successful ELISA pair to detect the spike trimer as the antigen. But my question is, what is the further purpose to choose trimer instead of the monomer? Is that for vaccine development?
Exactly. So the trimeric form, and I don't know if I made this clear enough. So the spike is actually a trimeric form. And as we saw, it goes through a series of cleavages, which is important for viral processing and activation, and what's known as priming, that allow it to be to allow the virus to be actually taken up into the cell. So of course, most of the neutralizing antibodies that we're seeing are directed against spike, and spike is normally in a trimeric form with the S1 domain on top, basically sitting, because that includes the receptor binding domain, receptor binding motif, and which sits on the S2 stalk. So being able to have a receptor, being able to recognize the receptor in its normal configuration, i.e. in the trimeric form, is of most value.
Oh, okay. Very insightful. Thanks.
Okay.
So, does anyone have the questions about today's presentation?
Oh. So POCT. So I'm assuming that's point of one person asked about POCT antibody kits. I'm assuming that's point of care, or basically, if that's what it is, and I'm sorry if I'm misinterpreting that. These would all be for in vitro use, of course. The kits that we're developing for nucleocapsid and for spike or be for in vitro use. And as I said, those pairs are being QA tested at the present time. And again, as Ying says, please, in addition to going to GeneTex, so that those questions will come to me anyway. If you want to email me directly at my to my email, I'm more than happy to address any additional questions you have, particularly anything related to products.
Okay, perfect. So since no further question at this moment, okay, thank you for your time today. And you just spare a few seconds to finish our questionnaire after you log out. So if any further inquiry, you can leave it that too. Or if you are willing to receive the presentation slide, please write down your email or contact our local distributor. So thanks for your participant and stay healthy and be well.
Thank you again.
Thank you.